we're going to go ahead and continue our discovery of the membrane here in this video by talking about the proteins which are found in the membrane, and those proteins are called membrane proteins. Okay, so there's actually two different types of membrane proteins. There are integral membrane proteins, and then there are peripheral membrane proteins. So integral membrane proteins are proteins which go through the membrane. Okay, they're integrated into the membranes, hence the term integral. Okay, so this in our diagram here, this protein would be an integral protein, as well as this one right here, because as you can see, they're actually going into the integral membrane. In contrast to that, you can have peripheral membranes, and peripheral membranes are peripheral proteins, and peripheral proteins are proteins that are kind of just hanging out on the outside. They're just connected to the very end like this. They don't actually go into, they don't penetrate the cell membrane. There's actually two types. There's actually a special type of integral proteins called transmembrane proteins. Let me write that down. Transmembrane proteins. And transmembrane proteins are proteins, integral proteins, which actually go through the entire membrane. Okay, so there's this would be a transmembrane protein because it's going across the complete width of the lipid bilayer. Now, the interesting thing about membrane proteins are they are also amphipathic, just like phospholipids. Now, we talked a lot about this in the previous video where we talked about membrane structure, how phospholipids are amphipathic, so you might want to go check that out if you haven't already. So, we know that this region of the lipid bilayer, the heads are hydrophilic. And then we know that this region of these are the tails of phospholipids. These are hydrophobic. So in the same sense, the proteins, the integral proteins which are found in the lipid bilayer also display this. So this region of the integral proteins over here, this region right here. Actually, let me use a different color. It's kind of hard to see. So this region right here of the integral proteins are hydrophilic, just like the lipid bilayer. And then this region right here of the membrane proteins, the integral proteins, are so all of this is actually hydrophilic. Okay, even the outside, because this, remember that this is all just H2O, right? So this region right here has to be hydrophilic as well. So anyway, so these regions of the integral proteins are hydrophobic. Okay, the reason for that is because if these regions of the membrane proteins were not hydrophobic and hydrophilic, then they wouldn't be able to fit well into the complete lipid bilayer, just like how the phospholipids have this hydrophobic and hydrophilic regions. Okay, so it's the integral proteins themselves are also amphipathic because they have hydrophobic and hydrophilic regions. So in terms of functions of transport proteins, in terms of membrane proteins, you can have transport. You can have transport is just moving materials in and out of the cell. We can have enzymatic activity, so that's basically helping reactions go faster. You can have signal transduction and cell recognition and intercellular joining, and these are all things that you'll learn more about in cell communication, and we're going to have a video for that, so just stay tuned. But just know this for now, all 5 of these functions are functions of membrane proteins. In terms of cell recognition, just as a brief definition, cell recognition is just when one cell recognizes another cell by identifying all these membrane proteins on the sides, right? If you have like maybe a membrane protein like this, and then there's of one cell, and then you have another cell which has another membrane protein, those two membrane proteins can interact, and then the cells can start to communicate with each other. Okay. in addition to that, for cell recognition, you can also have something called glycolipids and glycoproteins. Let's first talk about what glycolipids are. So glycolipids are basically carbohydrates, which are bonded covalently to lipids. Okay, bonded covalent bonding to lipids. Hence the term glyco, which is for the carbohydrates part, and then lipids, which is for lipids. Now, glycoproteins, you can probably guess what this is, but if you can't, it is just carbohydrate, which is covalently bonded to a protein instead of a lipid. Hence the term glyco for the carbohydrate part, and then protein for the protein part. Okay, and again, that is also a covalent bond. And glycolipids and glycoproteins also play an important role in cell recognition, rather than just these, in addition to these peripheral proteins right here. So, do membrane proteins move? Okay, so we saw in the previous video how the phospholipids have that lateral movement, which happens all the time. You know, back and forth movement like this between the phospholipids, and then you also sometimes have the flip-flop movement like this, which does not happen as often, but it does happen. So we know that the phospholipids are in constant motion. How about membrane proteins? Do membrane proteins also kind of move throughout the lipid bilayer? And the answer is yes, they do. These membrane proteins can move from side to side. They don't move as fast because they're massive, but they can move slowly from one side to another like this, kind of like move in laterally, okay? And there's actually an experiment which is conducted by these two scientists, Fry and Edidin, I think that's how you pronounce that, but they actually confirmed that membrane proteins do move. And this is an experiment that you should know, and you should be able to replicate for the AP Bio test, and make sure you pay attention. So what they did is they took a mouse cell, and then they took a human cell. I'm trying to stay consistent with my colors here. So they took a human cell and they marked the proteins with two different colors. So as you can see here, we have our mouse cell right here, and we have all the red marks, and then we have our human cell right here, and we have all the blue marks, and they just kind of pushed them together. Okay, the two cells were fused together. And eventually, what ended up happening is that we got this. They got this hybrid cell over here, and as you can notice, the membrane proteins are evenly distributed in the hybrid cell. Okay, we can see that it's not like there's one side. Like if you divided this into 1 and 2, it's not like one side of the hybrid cell has the red proteins and then another side has the blue proteins. They're all scattered about evenly. So that helped Fry and Edidin realize that the membrane proteins do move, and that the plasma membrane is indeed dynamic. Because if it wasn't for that, you wouldn't see all these proteins from the mouse cell and the human cell be completely dispersed evenly like this in this hybrid cell. Okay. Now, the last thing I want to talk about in membrane proteins is synthesis and secretion. Okay, so this is how membrane proteins are actually formed. Okay, so if you haven't already, you might want to go check out our video on the endomembrane system. There's actually 3 videos over there, and I talk about this process in that too, with the how the synthesis and secretion happens. You might want to go check that video out, but it's not necessary. You don't have to, because I'm going to talk about it again. So essentially what happens is that you have membranes which are synthesized in the smooth ER. The smooth ER is over here. What I highlighted was a nuclear membrane, but this is all the smooth ER. So what's going to end up happening is you get phospholipids. Synthesized by the smooth ER, and these phospholipids go and they go to the rough ER. In addition to that, you have, if you can look here, it's kind of hard to see, but there's all these ribosomes, these brown ribosomes, which make proteins. Okay, so for example, they have, if you see here, there's a orange protein which is made, and then there's also carbohydrates which are made from other parts of this the cell, which might go and come to the rough ER. So from there you can form a glycoprotein, because here this is a carbohydrate. Then you have the protein, which is made by the bound ribosomes. Go check out our ribosomes video if you haven't, if you don't know what this means. But they are created by their bound ribosomes in the rough ER, and then you have the phospholipids, which are coming from the smooth ER, and they all combine at the rough ER. Okay, now the rough ER is going to actually take all these materials and it's going to process them and create a vesicle right here. Okay, and this vesicle is formed through pinocytosis. If you don't know what that is, you might want to go check out our I think it's our bulk transport video where we talk about pinocytosis. You can also go check, we talk about what pinocytosis is in our endomembrane system videos, so you can also learn about that there. I don't want to get into too much detail on what that is. So through pinocytosis, we get this this vesicle right here. Now, this vesicle is going to go and it's going to fuse with the Golgi apparatus. So this part is the Golgi apparatus. And there's many processes that can happen. So here we have a glycoprotein, right? It might be hard to see because it's kind of small, but as you can see here, we have the carbohydrate, and then we also have the transmembrane protein. It's a transmembrane protein because it is going across the entire the entire width of the membrane, because the membrane is the light blue part. And if you look closely, you can see how the protein, the orange protein, is going across that entire layer. And then since we have that protein bonded to a carbohydrate, so this is actually a I don't know where I can draw this. I'm just going to draw it here. Different color. This is actually a glycoprotein. Okay, so the glycoprotein is going to come, and then what might end up happening is that the glycoprotein is going to go to the Golgi apparatus, and it might there's many processes which can happen. The glycoprotein might be broken down into just a protein here, and then a glycolipid here. This is a glycolipid. Okay, and then what will happen is another vesicle will form, and that vesicle will come and it'll go into this sac of the Golgi apparatus. The Golgi apparatus might have more than just two. It probably does usually have more than just two, but I have two here just for demonstration purposes. So many processes that can happen. You can the glycoproteins can be broken into glycolipids and just regular proteins. The proteins might combine with glycolipids to reform a glycoprotein again. As you can see here, we have a glycoprotein over here too, because we have the transmembrane protein attached to our carbohydrate right here. So what can happen is at the very end you get another vesicle right here, and if you notice here, our vesicle actually has three components. It has the protein here, it's a regular protein. It has a glycolipid right here, and then it has a transmembrane protein right here. Okay, and that vesicle is going to come and fuse. So if you look very carefully, what was inside the vesicle is now outside. I need a different color for this. What was inside the what was inside the vesicle is now going to be on the outside of the cell membrane, and what was on the outside of the cell membrane is going to be on the inside of the sorry, let me explain this again. I drew too many arrows. So again, what was on the inside of the vesicle is going to end up on the outside of the cell membrane, and what was on the outside of the vesicle is going to end up on the inside of the cell membrane. Okay, so if you can notice here, we had the proteins and we had the glycolipids. They are ending up right here on the outside, and then we have, if you look at these transmembrane protein, this transmembrane protein, the inside is yellow, and the outside of the transmembrane protein is yellow, and then the inside of the transmembrane protein is orange. So they kind of switch positions here, because now the orange part is on the on the outside over here, and the yellow part is on the inside. Okay, so that's just a generic rule that you need to know. The inside of the vesicle, what's on the inside of a vesicle is going to end up on the outside, and what's on the outside of the vesicle is going to end up on the inside. Okay, and I hope this diagram helps explain that. Okay, so that's all you have to know about membrane proteins. Stay tuned because there's much more to membranes than just that, which you'll learn in the next video. Membrane proteins are as simple as that.